I am Dr. Michelle Maddox. I'm a naturopathic doctor and a clinical consultant for Omni-Biotic, probiotics, and prebiotics. I have the pleasure of being able to interview Dr. Vanessa Stahlbauer today. She is an MD and is currently working as a professor in the clinical department of gastroenterology and hepatology at the Medical University of Graz and is leading the liver transplantation outpatient clinic at the Department of Gastroenterology and Hepatology. She is also leading the area of microbiome modulation for precision medicine at the Research Institute CB-Med. Her research interests include hepatology, acute and chronic liver diseases, liver transplantation, immunology, microbiome, intestinal permeability, and probiotics. Professor Stahlbauer serves as an editorial member and reviewer in several international reputed journals and has authored many research articles related to her research interests. And I am so very happy to be able to talk to her today. Professor, how are you doing?
I am fine, thanks.
So your work focuses mostly on hepatology, immunology, and the influence of the gut microbiome. It's such a fascinating topic, yet it's largely misunderstood by a lot of people. Would you begin by giving us just a little bit of an overview of how these functions, that liver, immune, gut, work together in a healthy individual, as well as the potential repercussions of imbalances such as dysbiosis or a microbiome imbalance?
Yes, of course. It's really true, as you said, that this is such a fascinating field. So when I started research in the field of hepatology, I started from the disease part, like seeing patients with lots of complications of liver cirrhosis. And then I started to be interested in immunology. And only at a little bit later stage, it became apparent that the gut microbiome influences these conditions to a large extent. So the gut microbiome, which is the sum of all the microorganisms that live in our intestine, plays a really large role in stabilizing the gut barrier. So usually when you're healthy, you have the bacteria that live in the gut. They produce a lot of helpful substances that can be used by the body, but they also keep the barrier tight. So the bacteria itself or also toxic products of the bacteria do not go via this barrier into our human body. But in the case of diseases, when the gut microbiome is damaged, or also the gut barrier is damaged, for example, by toxic substances like alcohol, this barrier breaks down, and then substances from the gut microbiome can enter our body. And we have a very unique situation between a communication between our gut and the liver. So all the blood that is in our intestine gets collected in a large vein that is called portal vein. And this portal vein directly goes to the liver. So the liver doesn't only get oxygenated blood by an artery, but it also gets this nutrition and other substance-rich blood via the portal vein. And also potential problematic substances like bacterial products can then enter the liver. And again, when you're quite healthy, then the liver can deal with that. The liver causes some inflammatory reaction, but then the problem is kind of solved. But in case you have a liver disease like liver cirrhosis, then these bacterial products can enter the circulation. This can be actually measured. When you draw a blood sample from a patient with such a condition, you, for example, find endotoxins circulating in the blood. And this can then cause systemic inflammation or even infections. And this then leads to clinical problems like complications of a disease. And this concept of the microbiome, the gut barrier, the immune system, and systemic effects is what we call the gut-liver axis.
So that was a really wonderful and succinct summary of the gut-liver axis and how it's impacted by our gut microbiome. I think it's going to be really helpful for the people who are listening to this. And I appreciate how well you drew that really complex interaction between so many different systems in the body into this kind of succinct little, this is what it is. It made it really approachable. So you mentioned that one aspect of your work looks at cirrhosis and how manipulation of the gut microbiome through targeted probiotics can positively impact liver function, as well as immune response and obviously gut microbial diversity and intestinal barrier integrity. You did a couple of studies. You did one in 2009 and one in 2020. What were the key takeaways of those studies? And were there differences between what you found in 2009 versus the 2020 study?
Yeah, so I'm working already since a long time on complications of liver cirrhosis. And I even did, before this 2009 study, also work in this field. Maybe I would like to start with this to kind of draw the whole picture. So initially, I was very interested in if the immune system works well or not in liver cirrhosis. And during a research stay abroad in London between 2006 and 2008, I did a lot of research on a certain type of immune cells, the so-called neutrophil granulocytes. This is the first barrier of defense that helps our body to fight pathogens. And we found out that these neutrophil granulocytes don't really work well in liver cirrhosis. And then we did our first study using a single probiotic strain that was given to these patients via a milk drink. And we saw a slight improvement in this immune function, in this neutrophil function. And when I then came back to Graz, I wanted to repeat the study in a larger setting as a placebo-controlled study in a large number of patients to prove this. And then I had the chance to work together with Institut Allergosan, and together with them, we developed a multi-species probiotic from data from cell culture experiments and other data using strains that are likely to improve the gut barrier, improve immune function, and thereby help to prevent complications. And so I planned and conducted this very large study. I was lucky also to get funding from an Austrian funding body for this. So we could include a little bit over 100 patients with liver cirrhosis into this study. And they were treated for 6 months with a multi-species probiotic, which is now already available on the market as Omni-Biotic detox. And we treated these patients for half a year and then followed them for another half a year. And the 50% of the patients got the true product and the others got a placebo. And in the end, we were very, very surprised that we were able to improve liver function during that time. That's really a very unique finding because usually you can't improve liver function in liver cirrhosis with any kind of treatment. You can improve liver function when you take away the toxic stimulus that causes liver cirrhosis, like when you treat hepatitis C or hepatitis B, or when patients stop drinking. But there is no drug currently available that can improve liver function. So we were very happy about this result. And we could also show that this multi-species probiotic improves neutrophil function and also monocyte function. But at that time, it was still very difficult in 2009 to look at the composition of the gut microbiome in such a large study with 100 patients, at least in my facility. So it took us a couple of years then, and then we could use the samples from the same study and did an in-depth analysis of the microbiome composition and the function. And we could also show here that the probiotic really improves the composition of the gut microbiome. So it's not only visible who has taken the probiotic. So we could find, of course, the probiotic strains in these patients, but above this, we could also find improvements in the general structure of the gut microbiome. So we found an increase in bacterial strains that are able to produce short-chain fatty acids, for example. And it is well known already that short-chain fatty acids improve the gut barrier. So this was then kind of the the proof for the clinical effect we saw in the first study, which made us, of course, very happy because we saw that it was not by chance, but really by modulating the gut microbiome.
It's very exciting to me when we have the ability to go back and reanalyze data with new technology. Technology, especially in the scientific community, is improving so very quickly. And so to be able to go back to that study and have an even greater understanding of the findings and see different findings that you just weren't able to see before, it's really awesome, and it's exciting to me. It's a great time to be in research, and it's a great time to be in medicine. 2 additional studies that you did, they further piqued my interest about how the microbiome impacts these particular systems. One of them looks at glucose metabolism and lipid markers in patients with diabetes, which is such a huge and prevalent issue across the world. What were your findings in that particular study?
Yeah, so in this diabetes study, we again did research on the same product, on Omni-Biotic Ketox, together with a prebiotic. The reason for doing this is that also in diabetes, we see changes in the gut microbiome, in the gut barrier, and in the immune system. So therefore, we came up with the hypothesis that the same product could be useful in this condition. And this was not only done by us, there are also 2 other studies, one from Poland and one from Saudi Arabia, who used the same product. We then decided to go for a similar treatment duration. So we used again 6 months of treatment, and it was again a placebo-controlled study. The patients took this probiotic and the prebiotic for 6 months, and then we also had some observation period afterwards. And when compared to the placebo group, we found some very interesting findings. We found an additional improvement in glucose metabolism. All these patients were diabetics and received, of course, their normal diabetes treatment. But in addition, we could improve glucose metabolism. We could also find some improvement in lipid metabolism and in the gut barrier. And what was most striking to me was that although we told these patients not to do any crash diets or change their eating habits or physical activities largely, so they should only carry on with their normal treatment over these 6 months, those who took the probiotic lost 2.5 centimeters of their hip circumference.
That's
so
awesome.
they didn't lose weight, unfortunately, but they lost fat. And that's, of course, a very interesting finding. So as I said, the patients, of course, had to do their normal treatment for diabetes or also for arterial hypertension. But it seems that probiotic treatment, in addition, can improve the effectiveness of these treatments. And I think this is really important in a condition like diabetes that affects millions of people worldwide.
Yes, that's a really interesting finding. And I read the study and I heard you say that they didn't necessarily lose weight. However, losing hip circumference is a really big deal. It is, this, at least in the US, we talk about this waist to hip ratio being such a key feature for increased cardiovascular risk and increased cancer risk. And so having a probiotic prebiotic regimen that would actually help take inches or centimeters off of a waist circumference and a hip circumference, that's a big deal, even if there isn't a weight loss necessarily with it. The other thing about that particular study that I found really interesting was there was a little bit of a discussion about a window of opportunity in this particular patient population. And we discussed that also with the pregnancy and with infants and toddlers. There's this window of opportunity for having a normal gut microbiome population. And it was interesting to see that term come up in this particular study. So the other study, one of the other studies that you did, it looks at the use of a targeted probiotic to offset the impact of long-term PPI use. Again, PPIs are everywhere in the United States. They're over the counter. anybody can buy them and start taking them. So a lot of people use them. And there's a huge number of people on them. It would be exciting to understand whether a simple supplement could be of benefit to this particular population. What did your study find?
Yeah. So maybe I'll start again a little bit before we did this study you're referring to see what actually the problem is. So we again reanalyzed data from our cirrhosis cohort. So one of my PhD students did this work, and she found out that when she looked at the gut microbiome composition, that one of the strongest factors that explained the variation between patients was whether they took a proton pump inhibitor or not. And then we looked at this in more detail, and we really found some shocking findings. We found that in patients with chronic diseases like with liver cirrhosis, when they have to take a proton pump inhibitor, this causes a certain type of dysbiosis. We find oral bacteria in the stool microbiome where they normally don't belong to. So everybody of us has bacteria in his or her mouth, which is perfectly fine. We need them there, but they usually, when we swallow them, but they are killed by the gastric acid. And if you don't have a lot of gastric acid because you take an acid blocker, then these bacteria can go further down and come to the gut microbiome. And again, when you are perfectly healthy, then this can be observed, but it's probably not a huge deal. But when you have a liver cirrhosis, these certain bacteria that cause this so-called oralization are associated with a higher risk of complications and even a higher risk of death. And so this was then the outset to say, okay, we need a solution, because many people have to take proton pump inhibitors. They can't be taken away, because otherwise they get acid-related diseases of the stomach and get complications from this. And so, of course, the first thought would be, why don't we use an antibiotic to get rid of two bacteria in the gut? But that wouldn't really help, because we would kill millions of other bacteria in there. So we had to come up with something else. So, of course, probiotics were next on our list to say that this could be a potential solution. And then we did our first approach. This is the study that you're referring to, that we already published, where we took a couple of probiotic strains together and treated patients with long-term proton pump inhibitor use. We found some very interesting findings, but to be honest, we were not completely happy with the result. So that's why this product is will probably not come to the market at any time, because we decided it was not good enough. We want more. But we could already show that with this product, we could improve gut permeability. So in those patients who had increased sonolin levels, which is a marker of gut permeability, they showed a very nice decrease when they took the probiotic. We also saw some improvement in lab tests of liver function. And what was really very interesting was that we saw an improvement in gastrointestinal symptoms. It is very interesting when you ask a patient who takes a proton pump inhibitor if they feel any side effects, they will hardly ever report anything. But when you then ask them to fill in a questionnaire, and after 3 months, we asked them to fill in the same questionnaire after they took the probiotic, we saw a huge increase in their gastrointestinal quality of life. So yes, we had some very good findings, but we were not happy enough with them. So what we are currently doing is searching for a better targeted probiotic. And we are already quite advanced with this. From a huge number of probiotic strains, we identified a couple of strains that are really targeted against this bacteria that cause this oralization. And now we work together again with Instituto Allergosan to make a product out of it and to also test it then in a clinical setting.
It's exciting that you're getting to develop product as you're improving quality of life. It's also always interesting to me when you work with patients, they report no symptoms, but then you hand them a questionnaire and suddenly they have a ton of symptoms on the questionnaire. So it's definitely a testament to how helpful questionnaires are when working with human beings. You mentioned that you're working on this particular study to develop a more targeted and a better product for this patient population. As knowledge of the gut liver expands and hopefully becomes as large as knowledge of the gut brain axis, I think there'll be a lot more interest in this. In addition to this study, is there anything else that you or some of your colleagues are working on or have been recently published that gets you really excited about this topic?
Maybe one topic that's really, really interesting and where we also start to work on now is that everybody of us inadvertently takes up microplastic particles when you drink something out of a plastic bottle, or when you eat something that was wrapped in plastic, or when you use toothpastes, for example. And there was already a study a few years ago from a colleague of mine, also a gastroenterologist in Vienna, who could show that actually this microplastic comes to the stool. So you find it in stool samples. This was very, caused a lot of media interest as well at that time. And that also caught my interest. And so I started a little bit of reading and found out that from animal studies, it's already known that microplastic causes changes in the gut microbiome. So when you feed mice with microplastic, they develop changes in the gut microbiome. And when you look at this data, it looks like the microbiome of somebody who is obese or has cancer or has some other diseases like diabetes. So you see a loss in diversity of the microbiome, and you see an increase in potentially pathogenic species. And on the other hand, there were also some studies completely outside of the medical field who showed that certain bacteria are able to degrade microplastic. So this was done at waste plants. So they collect bacteria from there, and they saw, oh, there are certain types of bacteria that can really get rid of the plastic. And what we are going to do now is to find out, first of all, whether microplastic really damages the human microbiome. And if yes, if we can find probiotic bacteria that can degrade microplastic. This would be really, really cool because we can't get rid of plastic. I mean, it's also has it's also not possible anymore to get rid of plastic as packaging material, and it also has some advantages because it's very light and you don't need such a lot of energy to transport things that are wrapped in plastic and so on. So it's not our goal to say that microplastic is bad, but if microplastic really damages the human gut microbiome, I would like to find out if there is a possible solution with probiotics to get rid of these negative effects.
That is really interesting and very exciting. I do read a lot about those microplastics, and I know a lot of people try to minimize plastic, but you're right. It's just not something that you can realistically completely avoid. And I never really thought about the environmental impact of making things all in glass, which is heavier and has, to transport and so on, has a potentially a larger environmental footprint. That's a really great point. I love that study. I'm really excited to see what you guys find.
So we just started last month with the study, so it will take some time, but yes, we are also very excited, and we really like this topic, and we think that this is something very important for the future.
Well, Dr. Stadelbauer, thank you so very much for your time and for sharing the findings of some of your studies and for giving us a heads up about studies that are coming that we should pay attention to. I very much appreciate your time and your sharing your knowledge and expertise with us this morning.
Thank you very much. It was my pleasure. And I hope that we will speak to each other soon in the future with some new exciting data. Thank you very much.
As do I. Thank you.